Welcome to the H3 Practical, which is about flow cytometry and protozoan grazing. So, you guys probably have not seen a flow cytometer before. The flow cytometer is just a big machine that essentially enumerates and counts. This one here is a S3E, and it's actually a cell sorter, so it has the capability to enumerate cells, but it can also sort live cells, which is really useful if you want to clean up a culture or split a culture into separate species, separate groups of cells. So it's really useful. So flow cytometry started back in the 1940s when machines were essentially set up with a laser light here just to capture a cell passing through the laser, and that was it. Wouldn't tell you any more about the cell whatsoever. Over the next 20 years, these machines developed quite rapidly, and they evolved into pretty much what you see today. So not only can they tell you how many cells are in your sample, they can give you an idea of size, granularity, autofluorescence, and you can use a multitude of dyes on your cells and samples which will highlight different features, and it will all be picked up with this machine. So they look at quite small cells. This one has a maximum cell size of about 30 microns, but works really, really well with sort of 10 micron size cells and smaller, and it's great for doing bacterial counts. So that's what they get used for mostly here. So flow cytometers are made up of three parts. They're made up of fluidics, optics, and electronics. And those three separate systems work together to determine how many cells you've got and what shape, size, fluorescence capabilities your cells have. So fluidics is basically this part here, and involves your sample. So you load your sample in here, it gets moved to the back, and you can see that that little tube at the back is where your sample disappears up and into the machine. It gets transported up through the flow cytometer in what is called sheath fluid, which is one here. The machine can put that sheath fluid under pressure, and different pressure means you will have a different width of sample stream. So on a low flow rate on the machine, it means that your sample is under high pressure, there's a lot of sheath fluid, and it's making your sample into a very narrow stream. So low pressure is really quite good when you've got a high number of cells, because putting that stream under high pressure, you can guarantee that every cell is going to be counted. We also have medium flow rate, and then we also have high flow rate. Don't tend to use medium flow rate much, but the high flow rate means that sheath fluid comes into the system. It's under low pressure this time, so you've got a very wide sample stream with a very small amount of sheath fluid down the edges, and it's transporting your sample up to the laser. And that's really good if you haven't got a lot of cells. So if you're talking about hundreds or maybe thousands of cells in your sample, it means that you've got a nice wide stream that goes up past the laser, and again it will catch every little cell that goes past. So the cells go up under these various flow rates. You can't see it, unfortunately, because it's contained in here, but there's a blue laser, 488 nanometer blue laser that comes across here, cuts here, and as your sample goes up, it passes through the laser. That's what we call an interrogation point, and at that point we're into the optic part of the machine. Your cell will pass the light, it will cut out the light for a few seconds, and as it passes through the light, we can set lots of different parameters, which you see on the computer here. I'll get onto those in a minute. Essentially, that cell cuts out, sends an electronic signal into the back of the machine, and it appears as a dot on the computer screen. It's not quite that simple. Once the electronic signal goes into the electronic part of the machine, we have all of these different features. Again, you can't see them within the machine, but they are what are called PMTs, so photo multiplier tubes. On the computer here, you can set up for all of these different photo multiplier tubes. So we actually have 6. We have a forward scatter, which is essentially the size of the cell. We have a side scatter, that's the light that comes off the cell as it passes through the laser, and that will give you an idea of how rough or smooth the surface of the cell is. And then we have fluorescence PMTs. We've got an FL1, which looks at your FITSI. We've got FL2, which is an orange yellow, so fibroresin stains. And then FL3 and FL4 are both red stains, with FL4 being a much deeper red and FL3 being the brighter, vibrant red. So they go to these detectors and the electrical signal is all sorted out and it comes up on the screen for you here as a dot. We tend to use dot plots, but there are other plots in the system. You can use a density plot, or you can even use a histogram plot, which will display the dots as peaks. Whichever is your preference, really. For the work that we do here, we prefer dot plots. Once you have your dots, you can put a gate around them, and you can work out how many cells are within that gate if you're going to enumerate. As I said before, the nice feature of this machine is that it's actually a cell sorter. So once that cell goes past the blue laser, before all the information gets sent to the PMTs, what we can do is we can gauge the population on our plots here, and we can ask them, or ask the machine, to sort the cells. And what happens is you can have a maximum of 2 gates, because one of the gates will apply a positive charge to the cell, and if you've got another gate, it will apply a negative charge. And when the cell goes up through the machine, the machine will recognise that any cell within that gate is one that you want to sort, and it will apply a positive charge. And if there was another gate up here, any within that gate are negative, and it will sort. And what it does is it sorts the cells via a fluid stream which comes between these 2 charged plates here at the back. One's negative, one's positive, and that charge drags your cell into tubes that are laid out in the bottom of the sorting chamber there. And essentially that's how it sorts. So again, you should have a handout with all of that on, to just go through essentially how the machine works and sorts. Okay, so now I've explained how the flow cytometer counts and sorts, we're going to run the practical. So, a couple of years ago, and only about 2 or 3 years ago, I would have done this with a microscope. You order a microscope, we would have been over in the teaching building, and I would have had cultures of paramecium and tetrahymenum, which are fairly large ciliates, and we would have fed them calming particles, which are basically crushed up bits of insects that are bright red, and the cells will quite happily eat them and ingest them and see them as food. And what I would have done was we would have had the 2 sets of ciliates, we would have given them both the same concentration of carmine, and it would have been a timed experiment. And we would have, over the space of 40 minutes, you would have taken samples at 0, 5, 10, 20 and 40 minutes, killed the cells with glutaraldehyde or formaldehyde, and then we would have put them under the microscope and counted the number of carmine particles within their food vacuoles, and then hopefully would have drawn a nice chart to show that ingestion rates increase with time. It's a bit long-winded, it's very high pressure for students, which we tend to forget. As technicians we do it all the time, it doesn't bother us, but it's quite stressful for the students. And because we now have Mike Subkoff here, who works a lot with flow cytometry and a lot with pico and nanoflagellates, the lectures have changed slightly, so we changed the feeding practical to fit with Mike's lectures, essentially. So what we've got today is, and you should have a handout again for this that you can refer back to, I have some cultures of a small flagellate called Paraphysomonas ovalis, and I do have a small video which will be part of the PowerPoint presentation which shows you these small cells. They are very tiny, they're about 10 microns in size. They are chrysophyceae, or they are golden brown algae as they are called, and they're quite a large group of protists, and they're abundant in freshwater, marine and brackish waters as well. So they're quite small, about 10 microns, and what we're going to do today is we're going to have a look at them and run them through the flow cytometer. Now I've already run a sample here, so it looks like nothing to you guys, but essentially where the green gate is that says P. ovalis, these are the golden brown algae. Everything below that and to the left is actually either rubbish, down here. This is likely to be very small particles of debris, maybe some bacteria in them. So these cells have been growing on rice grains, which explains all the junk down the bottom. And I've run this one this morning, but what I did the other day was I run this and I gated these cells here. I separated them from the rice grains that they've been feeding on, and I put a group of these cells in with another bacteria, and the bacteria is called Vibrio. And so what I did is Vibrio on a plate here. I just took a couple of these colonies and put them into solution, and I've been feeding these group of Paraphysomonas on this for about 4 days now. So these guys have gone from just feeding off of nutrients from a rice grain to feeding off of Vibrio. Now Vibrio is quite small, it's about 1 micron in size. So what I've got here is I have another bacteria. This is Cynococcus. And it's about 2.5 to 3 microns in size. So it's a bit bigger than the Vibrio. Vibrio doesn't have any fluorescence at all. It's a clear bacteria, and it will only show in the plots via size and light scatter. Cynococcus is a cyanobacteria, and it has a yellow-orange fluorescence. So it will show up really nicely on the flow cytometer. So the first part of this practical is to actually show you how the machine sorts. So I'm going to take some of the Paraphysomonas culture, and we're going to mix it with a little bit of Cynococcus, and you should be able to see 2 different cell groupings because of the size and because of the fluorescence. Then I'm going to get the machine to sort the 2 sets of cells, and we'll do an enumeration or a cell concentration calculation. The second part of the practical will be taking some of the Paraphysomonas that's been feeding on the Vibrio and feeding it Cynococcus, and we will set up a special chart on the flow cytometer to try to capture the Paraphysomonas consuming the Cynococcus. What we should have is, as they consume the Cynococcus, you should get an increase in fluorescence over time. And then we will run it again, and we'll run it with the Paraphysomonas that have just been feeding on the rice grains. Okay. So the question really is, and this works different every time we run it, the Paraphysomonas that have been feeding on the Vibrio Will they just be happy with the Vibrio and they'll just shun the Cynococcus and they won't bother eating it? Or will they drop the Vibrio and start eating the Cynococcus? Because they do prefer slightly larger bacteria. There's more nutrients, there's more, benefit from eating larger bacteria. So will they do that, or will they just carry on eating the Vibrio? And then for the second group, or the second set of cells which have just been feeding on the rice grains, again, will they be hungry? Will this influx of Cynococcus just make them eat? And will we get higher numbers over a shorter time frame of them eating these? Or will they just not be bothered? And as I said, it works different every time we've tried it. 3 or 4 times we've done this. So we will see. Okay. So what I will do now is I will take some of the Paraphystomonas that have been cleaned up and have just been feeding on the Vibrio. So in here I have my cleaned Paraphystomonas, and I'm just going to mix it a little bit because they do like to settle out and sink to the bottom of the chamber. So I'm just going to take 1 ml, put it into a tube. And then so what we put to the pipette here, and we'll put 200 microliters of Cynococcus into that. Should be more than enough. Well, okay, so in this tube I have 1 ml of the clean Paraphystomonas culture that's been feeding on Vibrio and 200 microliters of the Cynococcus. So we're not interested at this point in how they're going to feed. I just want to show you the sorting capability of the machine itself. So this goes into here. We load the system. So we push down. And like that. The machine will lock. And then over here, once all these features go bright green, it means it's ready and we can start. So I'm going to use a medium flow rate because I know there'll be fair amounts of bacteria, Cynococcus, but I also want to capture some of the Paramecium, Paraphystomonas ovalis. So let's go. And don't forget the Vibrio doesn't have any fluorescence and it won't show on the first cytometer. Okay. Ooh, very nice. So this will be the Cynococcus here. Very bright. And then somewhere up here, hopefully, will be the Paraphystomonas. Just let it run for a couple of minutes. So along the bottom of the screen here it will tell you how many events, total events the machine has counted. It will give you an indicator, it will tell you the time and also give, we're not doing sorting yet, but it'll also give us sort time as well. So I've set up lots of different plots here. I've got the forward scatter versus size. So this is size versus granularity. So we know the Cyanococcus are quite small, and we also know that they're quite sort of oval in shape, so they're very smooth. So this is why they're quite low on the system here. And our Paraphysomonas are a lot bigger, which is why they're going to be up there. And then I've got some other ones. We've got FL3, which is a red fluorescence against side scatter. We've got FL4 against side scatter. And then this one down here, which will be very useful, is FL2, which is your yellow phycoresin. So this is the fluorescence that the Cyanococcus give off. And then we've got FL1, which is any autofluorescence as well. So these two are the plots that we're most interested in. We've been running for just over 2 minutes now, so I will stop that there. Okay. As I said, I know these are quite small, and they're very regular as well. This is a gate that I put up earlier. If I just drag that down to the bottom, you see a tail coming up here. So Paraphysomonas are going to be here. They're about 10, 12 microns in size. And this is log, so down the bottom here will be about 1 micron to 2 microns, which is where our Cyanococcus are. So I'm going to put that gate back up there. Where the Paraphysomonas are. I'm just going to make it maybe a little bit smaller, and what I'm going to do is also rotate it. Okay, and we've got all these stats down here as well, so it's telling you how many events it counted in total, and it's also telling you how many Paraphysomonas we've got in that gate, which it says is 55 at the moment. What I need to do now is I need to draw a gate for the Cyanococcus, which I know are going to be here. And I'm going to use the FL2 versus size or forward scatter for my gates on this one. So I need to go draw a gate. So we'll highlight this plot here. Lips on it, so let's see, can I make it move it there. Okay, so on this plot here we've got our second gate, which is going to be the Cyanococcus. The cool thing about this machine is that you can actually take that gate and you can move to another plot, and you can actually apply that gate to this plot. It's just a tool that you can use to be really sure that the cells that you're capturing are the ones that you want. So let's just do this one, or this one's better. Do this one. So if I just right click on the mouse, it gives me the option to apply a gate, and I can choose now from P. ovalis or R2. R2 is the Cyanococcus. So if I pick P. ovalis, so it's cut everything out of that plot apart from this group here, which is that P. ovalis. I can clear the gate, and it brings it all back. And I can apply the second gate, which is the Cyanococcus, and we'll see what happens. Yeah, it's the Cyanococcus here. So I'm just going to clear those gates, and I'm just going to go down, and what I can do is also you can rename the gates. So I'm going to just put sins on this, so I know it's cytococcus. And if I wire that gate to this one, yes. The beauty is you can keep going and you can keep moving until you're happy. Okay, so I'm quite happy that this is the cytococcus here, and this is our paraphystomonas up here. So to sort these two, what I need is 2 tubes, one with the media in for the paraphystomonas and one with the media in for the cytococcus. Okay, so I've just rearranged the screen a bit here so we can see what we're doing a bit easier. So I've got these 2 dot plots next to each other. So this is the one where we've got our paraphystomonas gate, and this is the one where we've got our cytococcus gate. Right. In the machine, we've got our paraphystomonas mixed with our cytococcus still, and what I've got here is 2 tubes with the media, JN media for the cytococcus, and Volvic media for the paraphystomonas. So what I'm going to do is I'm just going to open the gate here, and I'm going to put this one on the left, that's the cytococcus, and I'm going to put the paraphystomonas media on the right. And what I'm going to do is I'm just going to close that. So now I'm going to use the software on the machine to sort these 2 sets of cells. So I'm going to right click on this and I'm going to select right sort. It's going to go into the right hand tube, and then I'm going to go click on this one and I'm going to click left sort. And then we'll click left sort. So you can see that the circles have changed colours. This one's orange and it's going to get sorted to the right. This one's green and it's going to sort the cells to the left. Before we sort, what I'm going to do though is I'm going to run the process for 2 minutes, and so we can have a go at making a calculation or enumeration about how many cells there are in the sample. What we can do is I'll run it for 2 minutes and we'll use the numbers here to do the calculation. Okay, so I'm just going to let it run for 2 minutes, and we're on a medium flow rate, which I believe is about 60 microliters in a minute. I will double check that. Just patience. Just coming up to 1 minute. With any more frustrators at the moment. Might have to refine that gate in a second and bring it down a little bit. 1 minute 50. Okay, so that stopped at 2 minutes and 2 seconds, but that's okay, that will go into the calculation. So we've got 307 cyanococcus cells here, and we haven't got any paraphysomonas at the moment. But I didn't expect there to be many, to be honest. It was a cleaned culture, so there's not going to be, and they've only had 4 days to feed and grow, so I wasn't expecting a huge number of cells there. So what we could do is run the old media again in a minute, just the paraphysomonas has just been feeding on the rice, and we can use that count. But for the moment, I'm just going to get this machine to sort cyanococcus. So I'm just going to pull that gate a little bit. Yeah, I'm happier with that. So that's actually jumped now to 575. This will all go onto another screen, so don't worry about that. So 575. So I'm just going to take a picture of that. So we'll just do that. We've got our 2 gates. One down just a little bit. Okay. And so we go sort logic. So this is telling the machine which way we want to go. So we'll have purity, so we'll have the cyanococcus going to the right with no limit, and then we'll have the ovalis going to the left, and I've put a limit of 100 on there, C. So what I'm going to do now is click go. It's okay. I can check, yeah. It's right sort because it's orange, and then this should be left sort because it's green. Yep. So we can run that again. And instead of clicking the start button, I'm actually going to click the start sort button, which sits next to that one. Okay, that's fine. So then we get this sort statistic screen comes up. Just move it over there for a second. So you can see it's counting how many cells. So left sort is our cyanococcus, this one is the P. ovalis. There's not much happening in that one. Just let this run for a couple of minutes until we get 100 in there. Okay, so I might let it run for 1 more minute, and then we'll stop. As I said, there's probably not many Parasimonus in this one because it was a cleaned culture. It's only been growing for a couple of days. So once I've shown you the sort feature for this, then I will put on the rice grain fed P. ovalis, and we will do a timed count again, probably for just a minute, and then we'll see how many cells we get so that you can do a calculation for numbers of cells in that one as well. Coming to the ends, we're at 2 minutes 50. We've got almost 2,000 cells. Okay, so I'll stop it there. So what I'll do now is I will take out the P. ovalis, which I didn't think got anything in that one. And then this is the cytometry. So you can see that there's little droplets on the side of the tube, and our cells may well be in those droplets. So I'm just going to very gently rotate the tube to catch the droplets. This is the trick to it. You don't want to end up tipping the cells out. So I've got them all there. So I'm going to take out the mix and open up the system. Yeah. You can't hear it, but it's just whirring, it's just having a clean. You can see on here that these are all greyed out at the moment, so it's not going to let me do anything until it's finished having a light little clean. Right. So what I'm going to do now is, so this is our JM media that contains the cyanococcus cells that we've sorted. So what should happen now is when I rerun this, most of this should disappear and we should start getting little dots in this gate here, which will indicate that it's sorted the cyanococcus from the rubbish, we hope. So I'm going to load that. I'm going to get rid of the sort screen, we don't want that anymore. And I'm going to run it on a high flow rate because although there's 2,000 cells in there, it's in about half a mil of liquid, so it's going to take a while. So high flow rate, which is about 100 microlitres a minute, and I'm not going to use the sort button, I'm just going to use the what we call the sample acquisition, and we'll see what happens. So it might take a while, but we should start there. There's a couple in there. You can see that big red dense block that we had there is almost gone. It's very low compared to last time, and we are getting cell counts within the gate here. The cyanococcus are quite small, so there will still be a little bit of dirt and debris, which is probably what this is here, but it's not as intense as it was before. So, and we're still getting counts within this gate here, and you can see them popping up now slowly over time. So it's coming up to a minute. So the last run was on a medium flow rate. You know, very dense population here of junk, and then cyanococcus were there. This time we're on a high flow rate, so it's much higher food flow through a sample. This is much reduced, and we've got our little cyanococcuses coming through on here, which is showing that the machine has sorted the cyanococcus into our new tube, which is great. So I'm just going to stop that now. What I'm going to do is, I did have a tube, so this is just P. ovalis on its own that's been feeding on the rice grains. So what I'm going to do now is I'm going to open this up. One. remove that. And I'll just run this for a minute on a medium flow rate so that we can get a count that you can calculate up to cells per mil. Okay, it's opened back up, so it's finished cleaning, so I'm going to clarify some motors on. I'm going to wait for this to go bright green, and again I'm just going to select fire and I'll leave it on a high flow rate. Okay, so I'll do it with fire. More of that, I'll just move that slightly back up there. So you can ignore this gate here. There's not any cytometry in there. This will just be rubbish and dirt that's collecting in here. And I'm just waiting for some of the cells to come into this gate here. What they might have done is actually settled out and stuck to the plastic tube. They do sometimes, so I'm just going to leave that running for a second and you can watch that, and I'll just do a new tube. Stop that one there. I'm going to try a new tube. Move some of these out of the way for the moment. Okay. This one. We'll try that again. So again, you can ignore this one. This one's just the side. There's no cytometry in it. Just clear all this. So let it run again. Just 1 minute, so let it carry on for 2 minutes and then I'll stop it. It's 1.5 minutes. So it's not showing up very well on this gate, so what I might do is put a gate on here, because it's showing up quite nicely on this one. So it's 2 minutes and 5 seconds. What I'm going to do is I'm going to put a gate here can see where it shows up on this one here. I've got a feeling it'll be this bit here. You'll see. So I'm going to highlight this. What I can do is I can here on gate. And there they are. It's handy. They've appeared all of a sudden. So the cells that I picked out on here as paraphysomonas are paraphysomonas. On this one here. So for 2 minutes and 5 seconds we've actually got 144 cells. And we'll do the calculation in a bit for that, and again I will take a picture of that one. Okay, so you've seen the machine sort, and we've got, well, I've got 2 screenshots now of the paraphysomonas and the Cynococcus after 2 minutes, and we'll go through how to make a calculation for numbers of cells per mil per litre in a moment. But we're now going to go on to the second part of the feeding practical, and what I'm going to do is I am going to check this sample. And I'm going to get a new tube with some new paraphysomonas in. This will be just the rice grain fed one, so they've just been feeding on nutrients that have leached out of the rice grain, and I'm going to introduce some of the Cynococcus. And then you will see, I'll just double check. Okay. A new tube. And what we'll do is we'll run this for half an hour. And we'll use a different plot this time, which I'll show you in a moment. And we'll run it for half an hour, and we will see, hopefully, if paraphysomonas want to eat or if they don't. So this in. So 250 microliters of Cynococcus in. Mix. And load this one into here. And we'll leave it on a medium flow rate. Start it off. So this time we're going to use this one here. We're just going to see what happens over time. So as the time passes, you should get little dots around here, and they will go up and up and up, hopefully, as they start to eat the paraphysomonas. You're going to get an awful lot of junk in this one because it's the rice grain one. Might not be very clear. We're going to leave it to run for 30 minutes, and we'll see what happens at the end. Okay, so now I'm going to repeat the process, and I'm going to feed the paraphysomonas that have been feeding on the Vibrio with a little bit of Cynococcus. We're going to use the same plot. We're going to run it again for about half an hour, and we'll see how many cells they eat, if they eat any at all. Okay. Right, well that's the end of the practical. It's a machine that's much better seen working in real time rather than this way, but unfortunately that's the way we've had to do it. There will be PowerPoints that go along with this, and hopefully it will all work, and I believe that there will be a question and answer session as well. And you can ask me questions, and hopefully we can get Mike to come along So if you have any questions about the cells, the morphology, the way they live and feed, hopefully Mike can answer some of those questions as well. If not, I'll just do my best. Thank you very much.